So what we would be trying is, there are some obviously this is a huge leap and has helped us in finding some of the those structures where we have the information before, but where there is no information, where there is no experimental structures available, and if there's a unique motif or protein, that is something that is still not answered by the AI based models. So some of the limitations that could be answered through experimental techniques, and we have those techniques which are evolving also at the same pace, and with the new structures and everything, I think this is going to be getting better and better only. The last database today that we're discussing about is the ChemiMBL. So ChemiMBL is another. Now we are discussing from discovery now to chemistry. So chemistry, ChemiMBL is a manually curated database of bioactive molecules that has drug-like properties. It brings together chemistry, bioactivity, genomic data as well, to aid the translation of genetic information into these effective new drugs. So ChemMBL is a database again which has two-dimensional structures, calculated properties such as log P, molecular weight, and then Lipinski parameters, and abstracted bioactivities such as binding constant, pharmacology, ADMET data. So this data is abstracted and curated from either publications like primary scientific literature, and covers fractions of these and helps us in structure activity relationships and discovery of modern drugs. So as we have been discussing and discussed so many these, not so many, at least four or five of these databases, it'll be really great if you can maybe browse some of them, as we will be also sharing with you the links and all after the session. So please, after the between the sessions, please go through this and have the questions that you can ask us related to them. Now we will be moving towards the case study, and we discussed about coronavirus and the SARS-CoV-2. So we'll take SARS-CoV-2 as a case study. So the entire all of the SARS-CoV-2 pandemic has also shown a light on many diverse contribution of this in silico structural biology work. So initially, structure prediction work we used to use we had used in that time modelers, ITESR and AlphaFold, and this DeepMind, all of that. But one of the key challenges now is to understand that you heard of the way some of these proteins replicate, and the way they create these mutations, and there are several copies. So understanding the way the mutations, actually these mutations can disrupt or become more lethal. So we have a case study planned on that, not in the first session, obviously, towards the second or third session, where we'll be able to look at the specific types of these variants and the structures, because many of the structures have now reported, and at a very good resolution. So we can do that case study where we can compare the variations that you could see in different types of these variants, where there are different mutations at different locations. So that is the case study that we have planned. But in addition, like if you look at the structures, all of this started with the spike protein, and which is at the surface of the coronavirus. So these spikes, and giving them their distinctive crown-like appearance in electron micrographs. That's the reason they are called they are called spike. So the spike initiation initiates the process of infection, and then binding to the receptors, and then fusing with the cell membrane to release the viral genome inside. And many of you might be are virologists and have a better understanding. But from a structural point of view, what I'm trying to describe here is that many other enveloped viruses, such as similar to spike proteins, to infect use the similar mechanism to infect cells. That has been known. For example, influenza and the enveloped glycoproteins of HIV-1 and Ebola as well. So the spike protein is composed of three identical chains. So we are talking about three chains, A, B, and C chains, and then together they form a complex with a small domain inside the virus, a membrane spanning segment, and a large ectodomain that extends the spike protein into different types of, like, it is very flexible to move in that manner. So in this slide, we I'm trying to explain the same thing, that when you look at these structures, and recent structures have revealed that the spike of SARS-CoV-2 and SARS-CoV are kind of flexible. So in this structures, the receptor binding domains are seen in different conformation. Now, about conformation is again one of the limitation. With the same protein, you will find different conformations. So how do you understand that what would be the best conformation to target, or what is the actual or the average conformation and all of that? So those are some of the questions that could be answered when you go through and learn from different aspects about molecular dynamic simulations, averaging structures, and understanding the movement. But actually these different conformations actually helped us a lot with all of this. The extended conformation is needed to bind the receptor, and and um, and the flexibility is a great advantage actually to the virus, because the researchers hypothesize that the recent variants are virulent because the receptor binding domains are particularly flexible, whereas the more widespread coronaviruses that causes the common cold, which are less flexible. So these are some of the examples that we should be able to take a look at, and let's do that in the context of visualization. So this last part of the session is doing some hands-on using molecular visualization. So that's, as we have shared that with you. So 6M17 and 6VSB are the 4-digit letter code that you can find on PDB. So let's click on some of these links and open this in a browser, and let me know if if you can see my browser or not. So this is a link that where we have opened a PDB file, and then in the context of visualization, we are going to use PyMOL. So PyMOL is a strong molecular visualization software, and in this slide you will be able to see that I have put together a lot of links from like how you can install PyMOL. So can you tell me, like, is it installed in your system, any of you? Like, do you know, like, have you installed PyMOL ever, or is it like something that you can do? So, all right. So many of you are saying yes, you have it installed, and for those who haven't or hasn't, I'll share these links in the chat. Perhaps that would be a quicker way. We will be sharing this in an email, but let me share all these links in the chat. So those who have not installed can utilize. And one of the thing is that PyMOL is a proprietary software by Schrodinger. But again, there are academic versions, educational versions available. They are a little tricky to install, but you'll find one of them in the link. So if you don't have the full version, or if you have the demo version, and if you use it for academic purposes, you can have the full version and all the capabilities, exploring some of these links. And I think that we will not do in the session, but if you have issues, please feel free to write that in the chat group, and we will be able to help you. Now, if you have installed it, the next step is to use it. And before using it, let's understand a little bit about the interface of PyMOL. So The interface, before understanding about the interface, it would be good to understand that what are the basics, like how do we visualize files and what is there in the pdb file. So can you tell me, like, do you know what is there in a pdb file, how the coordinates are stored, or do you have any idea like about the x-ray crystallography and all those methods that generates these experimental files? Because what I want to discuss next is this text file that we get from the experiments, like X-ray diffraction, X-ray crystallography, or other techniques. So this is the file. This is a text file. So when you're downloading from PDB, you kind of download a text file, and then you open it up on a in a software that kind of projects these coordinates in a three-dimensional space. Does that make sense to all of you? All right. So that's the whole idea, that from a text file, the software generates the coordinates and put it in a three-dimensional space. And I'm discussing this because it's important to understand, and if you understand this, you will be able to actually do some modifications on your own just through the structural visualization. So what we want to understand in this image that is on the left hand side of the screen is the PDB file. Yeah, we can do it, Girija. So Let's do this practically. I'll stop I'll go. We'll go to that website and let's do this there. So on the in the context of this file, this is a structure that I've been describing in the presentation. It's a membrane protein, and it's a viral protein. And there is this information that this is generated through cryo EM technology, and this is the resolution. Now, there are other information. There's an associated publication published in Science, so you can check the article here and understand more about it. That would be the best way to understand about the structure. And then there are different information. So this is the complete A chain, and D and C chain. So we have the sequence is 654 amino acid long, and this is one of the visuals of the structure. And then going down, you will find more information about different clusters, and then you can zoom in and look at these individual residues, compare these different sequence clusters which are similar to this protein, and then the second molecule. So this is like not just one molecule, there are two molecules. So angiotensin covering enzyme 2. So ACE2 receptor is also there. So you will be able to see the protein complex in this molecule. Now, similarly, there is a third molecule, the spike protein S1. So this information is right here, like the number of molecules that are there in the three-dimensional structure, co-crystallized. There are so many co-crystallized structures. Some of them are part of the experimental structure. Some of them are artificial artifacts from the solution. So you have to understand that which are the important part and which are not. And this sort of information is available right here. That's the way to read through this information here, that once you go to this page, read about this information, get that understanding. And there are here tabs also, so 3D view, and then annotation. So we can go through all of this. This will take a while, but let's download this structure first, and then we will do a visualization on PyMOL. And the next time, then when we meet, we will be able to maybe discuss all of this and answer the questions that you have, and feel you'll be able to ask those questions. So you will see that there are different ways that you can directly visualize right here in the on the browser. So you don't even need to have PyMOL for that. PyMOL is for those maybe where you're predicting and you're modifying. So it's great to have PyMOL capabilities, generate high resolution structures. but here also these capabilities are there. You can look at active sites, you can look at different sites, you can click and understand the interaction also, which is a great interface that PDB is now providing within this portal. So I wish that, I hope that you all are going to utilize some of these links, and I'll put it in the chat, especially this one that we are looking at, so that you all can look at it, and we can maybe do it together. So now let's download this structure. So while downloading these files, you'll find different file formats that are there. Now, PDB format, there is this MMCIF format. So we can discuss about these formats, but let's look at some of the one of these formats. So I've downloaded this file, and I can open it in a text editor. So let me open this file in a text editor. So you can use Notepad, Notepad++, WordPad, any of these text editors to open a PDB file that you have downloaded now. So using the link, as you saw, that I easily downloaded that file. And this is how it looks. Like, this is the structure file. This is the maybe a receptor file. This is how it looks. It has a lot of remarks from the top towards going towards the end, and these remarks are not read by the software, but they explain the characteristics of this molecule or the structural factors, and some of the chains, some of the interactions, as well as these sort of information. So there's a lot of information this gives. Just to tell you in a nutshell, so header, title, author records provide information about the researchers who defined the structure. Numerous types of records are available to provide other type of information. Remark record provides contains free form annotation, and then the sequence res records give the sequence of these three peptide chains like A, B, C, D, and all. Atom records. Now, what is important here is the atom records. From here, this is the important section. This is the actual file. This is the actual structural file. So this is atom. This atom describes the coordinate of the atom that are the part of the protein. For example, the first atom line describes the alpha N atom. So this is the nitrogen at the alpha position of the first residue of the peptide chain A. I hope that this is kind of clear when we will look at it in a three-dimensional visualization as well. But that is how it is. So it is a valine, and the residue is valine, and the first three floating points. This is X, Y, Z. This is X coordinate. This is Y coordinate. This is Z coordinate. So X, Y, Z will be transposed or will be plotted in a three-dimensional software called PyMOL. Now, I think this is clear, and this is kind of, I explained it in much more depth. The next three columns are occupancy, temperature factor, and the element name, respectively. Then you will find towards the end, there are different chains. Like, I'm scrolling, and I hope all of you are able to see this. So this is A chain, as we know that this is a big protein. Then there is B chain. You see that A has changed to B now. Now this is B chain, so another subunit, and then there will be other chains also. E chain and C chain. As we were reading the molecule there, D chain, and then after the atoms, I just wanted to show heteroatoms. These are the heteroatoms. Now, the ligands are usually heteroatoms. So here you will find these NAG, which is like a crystallized, I think these NAGs are present. These are, sorry, not crystallized, like there are some conditions for this in the solution. And then they are there. And then there is water molecule, and there is zinc also. So here towards the end, you will find zinc, water molecules, and all of that. So each chain had some water molecules that have been traced and put together here. So this is how you read the PDB file. Now you can manually actually extract some of this information and paste it in a new file and create .pdb. So you can simply, like, if you want to extract the like the F chain, you just cut it like properly and paste it in a same format and save it, like, and rename it like fchain.pdb. So that way you can manipulate this PDB file. You can take some of the segments and open it and do those exercises. And those will be the assignments for today's session, to manipulate my PDB file and to do this on your own. Now, the next thing we are going to try is PyMOL. Now, I have installed PyMOL on my system. Now, I'll open PyMOL by this command, PyMOL, and this ampersand sign is actually a sign that let me use the console. Now that's not important. So yeah, PyMOL, that is what we would be interested in. So we have opened PyMOL and we have downloaded a structure. Now let's open that structure in PyMOL and let's look at it and understand what are the different ways we can visualize it. So now I have opened it in PyMOL and I'm rotating it using my key mouse pad, tracking pad. Now let's yeah, I think it's visible to all of you. Now here there are a few commands, ASHLC. So A stands for action, and there are two tabs here, all and this one. So you can actually load multiple structures, and in the next session we will be discussing about structural alignment, structural comparison, and there is a workshop also coming up on AlphaFold and doing all of this. So I hope that as many of you would join the program, will be coming and participating on all of those events. Now looking at the structure, now you can zoom in as much as you want, and you can dive and look into each molecule. So here I've just selected, clicked here, and there's one atom which is selected. Now you can see that there are dots, but it is not showing in the presentation because the presentation that we are looking at is the cartoon presentation. Now there are different types of presentations, visual presentations that we will be looking at, that we will be exploring, but this is a cartoon presentation. Now to look at the side chain, you need a stick presentation. This is a stick presentation. So this is the NAG molecule which is plotted all over here, which is you can see all over the place, NAG molecule. But I have clicked on an amino acid which has a ring, as it seems. Now I want to see what is this amino acid. So what I will do, I'll go to the select, because I've selected it. There's a new tab that has come up which says SELE, which is select. This is selected. Now whatever I'll do will make change to only this area. So this is the select, and I'll do what? I'll try to show stick. As I was telling you, this could be seen in a stick representation. Now you can see it's a stick representation, and you can look at the residue. You can take a look at the residue. Now, this is important in the context that if you want to know about that, if there is a mutation and you have figured out that you know that there is this mutation, so you want to see what is the effect of the mutation, and you can check out that by looking at the residue, and then selecting that residue like it is selected. And in action, I'll use this function called modify around 4 angstrom. So I wanted to see that where this residue is interacting with, is it interacting with any other intramolecular interactions within the protein. So let's look at that, because if we want to change this, if we want to mutate it, obviously some of these interactions will be lost. So as I did that, what I did is that I went in action, modify around 4 angstrom. So I checked what are the interaction around 4 angstrom. Now I have other residues also getting highlighted, or there are these selections. Selection has expanded to other molecules, which means that there are other molecules that are there interacting. Now I can also manipulate the structure and mutate it. This is what, let's take a look at what is this what is this side chain. So to name it, there is this tab that says label. So this is label. Here you can also written action is written, show is written, hide is written. So you can show something, you can hide the same thing. This is label, it's written, and then there is color. You can change the color to different colors of your liking, or to highlight it basically. Now I have selected this. Now I want to mutate it, but before that I need to know which atom is it. But this is tyrosine. This is tyrosine because you can see here, when I've selected, this console is also getting updated, and it's telling me that this is a B chain tyrosine at 641 position. But we can label it, and she see it in here. So we label it. It's showing that now in the screen. You can see it's so showing TYR 641. Now I want to change this tyrosine to alanine. How would I do that? How can I do that in PyMOL itself? So there is this plugin or wizard. There is this mutagenesis that you can do here, right in here. Right, it comes up here. Now I have selected this one, and I want to change this mutation. I want to bring a mutation in the structure by changing tyrosine to alanine. Now what do I want to change it into? Alanine. So I'll put ALA, and I'll apply. So I've changed mutate to alanine, and these are some of the other options that I can change. So I'll apply. It's asking me to pick a residue. So let's pick a residue. All right. So let's try this again. It's asking me to select a rotamer. Okay, so I think there is a library file for rotamers that we have to put in here, but there is this mutation. I think this now has changed into alanine. So the side chain has gone, but I think with the rotamer library, it will be like give us probabilities of this mutation. So now this mutation it's changed into alanine. So this is one of the way I was just showing all of you to work on these protein structures and do some manipulation using PyMOL. Now, what we did is that we downloaded a structure, we played around with it, and we learned about some of these things. Now, for the assignment, for the students or participants of the program, we'll be sharing some of these protein IDs and ask you to prepare a report where you have done some of this work, or we can discuss any of your interest for what we have covered in this session. Now, as we learned about PyMOL, we learned about PDB, we learned about different databases, and we also learned about how can we do some of these manipulations. Now, we can go much depth of these, and we will be using these tools throughout the program. This is just an overview so that many of you get started with these things and develop these expertise throughout the program with the help of the mentors. Now, going back to the presentation. So in PyMOL, what we have done is we have done the installation, and then we have done some basic analysis. Then another visualization software that is there is Chimera. And in the next session, we will look at Chimera and we will look at some of the capabilities of Chimera, and where which is utilized for. Similarly, we will do mutagenesis, and we will do some RMSD calculations where we will compare two structures and see how much they are different. And then we have these sessions coming up: introduction to cheminformatics in the next session. Then we have chemistry and protein databases, many of which we have covered, but we will go in more depth. These are the some recorded sessions also. And then we have the protein structure prediction, some of the case studies like docking experiment setup. What are the different, as we were discussing, more the more we are being able to mimic it, the more high probability of this to be accurate. And then the drug repurposing case study that we were discussing will be discussed as well. And then the advancements of transformation using GPU computing and deep learning, and different molecular generation using deep learning. So these are some of the things that we have it put together. And before I pass it on to Sonalika to discuss about the other aspects, I would request you to join LinkedIn, this group. This is a group where this has grown now more to more than 20,000, 2,000 people. So here we frequently post about the job opportunities and all of that. And in the OmicsLogic group on Facebook, we also do the same. So if you're on Facebook, LinkedIn, please join these groups, and then you'll be able to find job opportunities there, as well as on the Learn OmicsLogic portal. So that's what I wanted to cover in today's session. And I'll be happy to take the questions that are there in the chat, if Sonalika, or you can let me know about those questions. And in the meantime, it was great meeting you all for the first session. I hope that this was something that was helpful and you learned something new. And we will try to make this more and more interesting throughout the program. Thank you so much.